Hello, my name is Zane Fligenhart, and today I'll be presenting on yellow fever virus. Looking at a historical perspective of yellow fever, scientists believe it originated in East or Central Africa roughly 3,000 years ago. This is indicated by phylogenetic analysis and genome sequencing. Throughout the 15th and 16th centuries, Africa has seen many outbreaks. However, natives had some form of acquired immunity that mitigated their symptoms and made it more similar to an influenza infection. However, Europeans who had not been exposed to yellow fever before had worse symptoms and much higher mortality rates. It is believed that yellow fever virus was able to spread to the Western Hemisphere through the Columbian Exchange, specifically the slave trade. Looking at outbreaks in the New World, the first outbreak in the New World happened in 1647 on an island of Barbados. The first time the disease was described as yellow fever was in 1744. Dr. John Mitchell described the outbreaks as yellow fever in Virginia. It is believed that sugar plantations and deforestation contributed heavily to the increasing mosquito population that caused yellow fever at this time. One of the most infamous yellow fever outbreaks was in 1793 in Philadelphia. During this time, thousands of individuals died, equating to roughly 9 to 10% of the population of Philadelphia. It was so severe that the national government fled, including then President George Washington. During the 18th and 19th century, there were over 25 major outbreaks that were recorded. It is believed that it is a much higher number, but these cases were not all documented. Looking at the discovery of the virus and how it's transmitted, the U.S. had invaded Cuba during the turn of the 20th century, and Surgeon General sent a pathologist and bacteriologist named Walter Reed into Cuba to investigate the disease that was killing thousands of American soldiers. One of his associates named James Carroll allowed himself to be infected by a mosquito, and he subsequently developed a very severe case of yellow fever. Luckily, he survived. However, this proved that a virus that was transmitted by mosquitoes was likely the causative agent. However, it wasn't until 1927 that Adrian Stokes isolated the virus from a man in Ghana. Examining the viral structure, the virus is quite simple, yet very effective at what it's programmed to do. It is a positive sense single-stranded RNA virus. This essentially means that its genome can be directly converted from RNA to protein. It doesn't need to be copied into other forms to be used as instructions for protein construction. It's 40 to 50 nanometers in width, which means it cannot be observed under a light microscope. It's an enveloped virus, as you can see from the diagram to the right, and three main proteins serve crucial functions inside the virus. The function of the virus's E proteins is to attach the virus to receptors on the host cells. They initiate the biggest immune response from the host. The M proteins appear to keep the E proteins functional during the assembly of new viruses, but M proteins have been studied a lot less than E proteins. C proteins are located in the virus's nucleocapsid, but scientists aren't exactly sure what their main function is. When discussing transmission, it is important to know that mosquitoes are the primary vector for this virus. Haemogogus species are responsible for most of the transmission in South America and many cases in North America as well. In Africa and also in some cases in the Western Hemisphere, Aedes mosquitoes are also known to carry the virus. However, female mosquitoes are the only ones who are bloodsuckers who can actually transmit the virus. There are three main cycles involved in the transmission process. The first is the jungle cycle, which involves transmission of the virus between non-human primates, such as monkeys, and mosquito species found in forests. The virus is transmitted by mosquitoes from monkeys to humans when humans are visiting or working in that area. In Africa, an intermediate cycle exists that involves transmission of the virus from mosquitoes to humans living or working in areas that border densely forested areas like jungles. In this cycle, the virus can be transmitted from monkey to human or from human to human via mosquitoes. The last cycle, known as the urban cycle, involves transmission of the virus between humans and urban mosquitoes, primarily Aedes mosquitoes. The virus is usually brought to the urban setting by a viremic human who is infected in the jungle or the savanna. Prior to replication, the virus must be able to enter the host cell. Those E proteins we discussed earlier attach to human cell receptors, specifically hepatocytes. The hepatocytes then bring the virus inside through an endosome, which is a membrane-bound vesicle. The virus is released from the endosome by fusing its own membrane with the endosomal membrane. And through this mechanism, the viral genome is then released into the permissive cell where it can replicate. The replication cycle is depicted as a series of regulated steps. In the first step, it's the translation of the polyprotein. Second is the co- and post-translational processing to produce structural proteins and non-structural proteins. The third step is the synthesis of complementary negative sense RNA by the RNA-dependent RNA polymerase and other viral replicase components. In the fourth step, the synthesis of progeny genomes by transcription of the negative strand. And lastly, in the fifth step, progeny genomes are packaged into nucleocapsids and bud intracellularly to acquire their envelope. When an individual human is bitten by a female mosquito, they are intradermally inoculated with anywhere from 1,000 to 100,000 viral particles. The virus then begins replication in dendritic cells that are locally in the area where they were bitten, and then it spreads to the lymph nodes. Utilizing the highly connected lymphatic system, viral particles then are able to spread to other organs. The three most prominent sites of viral reproduction are the liver, the spleen, and the lymph nodes. And in severe conditions, the body can actually go into circulatory shock from the high cytokine levels that result from the viral infection of many host cells. Yellow fever virus has three distinct stages of infection. The first being period of infection. So roughly the incubation period is anywhere from 3 to 6 days, but once you see symptoms, they're very rapid onset. These symptoms include things like fever, headache, photophobia, which is fear of light, nausea, vomiting, loss of appetite, dizziness. It's very similar to the symptoms of influenza. It can actually be mistaken for it. In most cases, patients will transition into a remission period where fever and other symptoms subside for up to 48 hours, and oftentimes people will begin to recover at this point. But about 15% will actually transition into the third stage of the virus, where their symptoms will recur but be much worse. In this third stage, known as the period of intoxications, usually occurring 3 to 6 days after your initial onset of symptoms, the symptoms get much more severe. You get your return of fever, nausea, vomiting, abdominal pain, and jaundice, which is where it gets its name, yellow fever. Organ systems are heavily impacted by this. You have things like renal failure, hemorrhage, myocardial bradycardia and myocarditis, and central nervous problems, which can cause convulsions in patients. And there is a possibility of death. Mortality rates are under 10%. However, people who reach this stage of the disease are at severe risk for death. Unfortunately, no cure currently exists for yellow fever virus. Antivirals are in development. One of the ones noteworthy is ribavirin, which is still being tested extensively to test its effectiveness on yellow fever patients. Instead of treating patients, supportive care is usually recommended, which has a high likelihood of increasing survival rates. This can include things like rehydration, nutrition, FFP for clotting, dialysis, and treatment of hypotension. Instead, scientists and doctors and other healthcare professionals focus on prevention of yellow fever virus and its spread. The first method is through a live attenuated vaccine, which we'll discuss a little more later, but that does help give immunity to people who get the vaccine before they're exposed to yellow fever. In one study done by the World Health Organization, 99% of people who got the vaccine had some form of immunity to the virus after 30 days. The next is through vector control, specifically targeting mosquitoes and lowering their population numbers in areas where there are a lot of people at risk for infection of yellow fever virus. And lastly is epidemic preparedness. in which countries need to be more prepared in their ability to produce the vaccines and also uh, isolate cases. Major problems associated with yellow fever virus are a lack of knowledge about it and our preparedness to fight it off in case of an epidemic. The virus exhibits polymorphism with high mutation rates, meaning that there are many different strains and they vary quite a bit. Major differences between the vaccine strains and wild type have already been identified. For example, surface proteins that help with attachment, like the E proteins we discussed earlier, are much different in wild types than they are in the vaccine strain, making the vaccines less effective. Also, molecular mechanisms of immunogenicity and pathogenicity in vivo are completely unknown. There are shortages of vaccines in areas where they are needed most, like South America, specifically Brazil, and also Africa. Increasing global temperatures are expanding the range of which the vector, like mosquitoes, can spread into new areas, drifting further away from the equator and creating more susceptible targets. Lastly, replication of viruses in tissue cultures is not a good, accurate representation of an infection in a human. Tissue culture is slow, has a long latent period, and there's only moderate titers of the virus are produced. And host cell protein and RNA synthesis are not shut off like they are in actual infected individuals. I hope you enjoyed this presentation on yellow fever virus.